In 2016, the Nobel Prize in Physiology or Medicine was awarded for the discovery of a cellular process that has been happening in your body every day for your entire life. A process that your cells use to clean themselves, repair themselves, and extend their functional lifespan. And that you can activate deliberately by simply not eating for a specific period of time. The process is called autophagy. The scientist who characterized it is Yoshinori Ohsumi, and the mechanism he described explains why the oldest documented intervention for human health, fasting, produces biological effects that researchers are only now beginning to quantify. This is the story of what autophagy is, what triggers it, how long you need to fast to activate it meaningfully, and what the research on fasting and biological age actually shows. Every cell in your body accumulates damage over time. Misfolded proteins that the cell cannot use, damaged organelles, particularly mitochondria, that produce reactive oxygen species instead of energy, viral particles and bacterial debris, aggregated protein complexes that interfere with normal cellular function. The cell has two primary strategies for dealing with this accumulation. The first is the ubiquitin proteasome system, a molecular shredder that degrades individual damaged proteins. The second is autophagy, a larger-scale process in which the cell engulfs portions of its own cytoplasm, including entire damaged organelles, in membrane-bound vesicles called autophagosomes, and delivers them to the lysosome for degradation and recycling. The word autophagy comes from the Greek for self-eating. The cell is literally consuming parts of itself, but selectively, targeting the damaged and dysfunctional components while preserving what works. The degradation products, amino acids, lipids, nucleotides, are released back into the cytoplasm and used for new protein synthesis and energy production. Ohsumi's contribution was identifying the specific genes, ATG genes, for autophagy-related, that control this process, characterizing their proteins and their functions, and establishing autophagy as a regulated, genetically controlled process rather than a passive cellular housekeeping mechanism. This genetic foundation enabled the subsequent explosion of research on autophagy in aging, cancer, neurodegeneration, and immunity. The critical insight for human health is that autophagy is not a constant, always-on process. It is regulated by the cell's nutritional and energy status. It is upregulated when nutrients are scarce and downregulated when nutrients are abundant. The cell cleans itself when resources are limited and builds when resources are plentiful. This regulation is mediated by two master metabolic sensors, mTOR and AMPK. mTOR, mechanistic target of rapamycin, is the cell's primary nutrient-sensing complex. When amino acids, glucose, and growth factors are abundant, mTOR is active. Active mTOR signals growth. It promotes protein synthesis, cell division, and anabolic processes. It simultaneously inhibits autophagy. A cell in growth mode does not clean house. AMPK, AMP-activated protein kinase, is the cell's energy sensor. When cellular energy is low, when the ratio of AMP to ATP rises, indicating that energy stores are depleted, AMPK activates. Active AMPK does the opposite of mTOR. It promotes energy production, inhibits energy-consuming anabolic processes, and activates autophagy. These two kinases function as a switch. High nutrients, high energy, mTOR on, AMPK off, autophagy suppressed. Low nutrients, low energy, AMPK on, mTOR inhibited, autophagy activated. Fasting shifts the switch. As blood glucose falls and amino acids are no longer entering the bloodstream from digested food, mTOR activity decreases and AMPK activity increases. The cells shift from growth mode to maintenance mode. Autophagy is activated. The timing matters because the metabolic shift is not instantaneous. Blood glucose and insulin decline gradually after the last meal. Liver glycogen, the body's immediate glucose reserve, is depleted over approximately 12 to 16 hours of fasting, depending on activity level and metabolic rate. The activation of autophagy tracks this metabolic transition. It begins to increase meaningfully as the fed state fully resolves and the fasting state is established. Research measuring autophagy markers in human blood, particularly Beclin-1 and LC3 proteins, finds measurable increases beginning at approximately 12 to 14 hours of fasting, with more robust activation at 16 to 18 hours and continuing to increase with extended fasting. This is the biological basis for the 16-hour fasting window that has become the most studied intermittent fasting protocol. 16 hours without food, 8 hours of eating. It consistently falls within the window of meaningful autophagy activation without requiring the extended multi-day fasting that produces significant metabolic stress. The relationship between autophagy and aging is direct and mechanistic. The hallmarks of aging identified in the research literature, including accumulation of damaged proteins, dysfunctional mitochondria, genomic instability, and cellular senescence, are all conditions that autophagy specifically addresses. Efficient autophagy clears damaged proteins before they aggregate. Mitophagy, the selective autophagy of damaged mitochondria, removes the mitochondria that produce reactive oxygen species, replacing them with healthy mitochondria through biogenesis. These are not indirect or correlational relationships. They are the specific substrates that autophagy degrades. In model organisms, genetic manipulation that increases autophagy extends lifespan. In yeast, worms, flies, and mice, upregulating autophagy genes produces animals that live longer and show less age-related functional decline. Conversely, reducing autophagy accelerates aging phenotypes. These are causal relationships established through genetic experiments, not correlations. In humans, direct manipulation of autophagy through genetics is not possible, but the fasting protocols that activate autophagy have been studied in the context of biological aging using epigenetic clocks. Epigenetic clocks are measurement tools based on the observation that DNA methylation patterns, chemical modifications to DNA that regulate gene expression without changing the sequence, change in consistent and predictable ways with aging. By measuring methylation at specific genomic sites, it is possible to estimate biological age, the age of the cells and tissues based on their molecular state, independently of chronological age. Biological age can diverge from chronological age in either direction. A 60-year-old with a biological age of 50 has cells that function like those of a younger person. The same person with a biological age of 70 has cells in an accelerated aging state. Fasting protocols have been studied using epigenetic clocks. A study published in Nature Aging used a rigorous clinical trial design to measure epigenetic age before and after a 5-day fasting protocol in older adults. Biological age decreased by an average of 2.5 years during the fasting period, with the reduction persisting for months after the fast ended in participants who maintained some degree of time-restricted eating. This is not a permanent reversal of aging. It is a measurable shift in the molecular state of cells in the direction of a younger biological age, a shift produced by the same metabolic mechanisms that autophagy research has characterized at the molecular level. The practical question is what fasting protocol is sufficient to produce meaningful autophagy activation without the adverse effects of extended fasting? The evidence supports a graduated view. The minimum threshold for meaningful autophagy upregulation in human cells appears to be approximately 12 to 14 hours without calories. This is achievable for most people through time-restricted eating. Finishing dinner by 8 p.m. and not eating again until 10 a.m. produces 14 hours of fasting without requiring any skipped meals in the conventional sense. The 16 to 18-hour range produces more robust autophagy activation based on the biomarker data available. This is the range of the 16-8 protocol. 16 hours fasting, 8 hours eating. That is the most studied intermittent fasting approach in the literature. Extended fasting beyond 24 hours produces additional autophagy activation and additional mTOR suppression, with some evidence of more significant cellular renewal effects. But it also produces more significant metabolic stress, is more difficult to sustain, and introduces considerations around muscle protein turnover that are relevant for people concerned with maintaining muscle mass. For most people seeking the longevity and cellular maintenance benefits of autophagy without the complexity of extended fasting, a consistent daily window of 16 to 18 hours without calories, combined with adequate protein intake during the eating window to support protein synthesis when mTOR is active, represents the most evidence-supported practical approach. Coffee and tea without additions do not meaningfully suppress autophagy during the fasting window. The small caloric content and the absence of significant insulin stimulation preserve the fasting state. Black coffee may actually enhance AMPK activation through its effects on catecholamines. The boundary is meaningful caloric intake, particularly carbohydrates and protein, which are the primary mTOR activators. Here is the synthesis. Autophagy is a genetically regulated cellular cleaning process that degrades damaged proteins, dysfunctional organelles, and molecular debris. It is activated when mTOR is suppressed and AMPK is active, the metabolic state produced by fasting. Meaningful autophagy upregulation begins at approximately 12 to 14 hours of fasting and increases with fasting duration. The hallmarks of aging that autophagy addresses: Damaged protein accumulation, dysfunctional mitochondria, cellular debris are directly causally related to the aging process in animal models where autophagy manipulation alters lifespan. Epigenetic clock research in humans shows measurable reductions in biological age following fasting protocols, reductions that persist beyond the fasting period in people who maintain time-restricted eating. The practical minimum is 12 to 14 hours without calories daily. The most studied and most practically sustainable protocol is 16 hours fasting and 8 hours eating. Extended fasting beyond 24 hours produces additional effects with additional complexity. Your cells clean themselves when you stop feeding them. The Nobel Prize was awarded for characterizing exactly how. The timing is 16 hours. If this changed how you think about the hours between dinner and breakfast, hit the like button.
Share it with someone who thinks fasting is extreme, and subscribe